Hello, guys. Welcome to another high yield pathology series by PathoGeeks. This is Dr. K. Shravani. Today, we are going to start with apoptosis. It is a genetically programmed cell death and it is energy dependent. That is, it uses ATP. Usually occurs in a single cell or small group of cells. Mostly physiological. Can be pathological as well. So the physiological causes include, removal of supernumerary cells, involution of hormone-dependent tissues, removal of cells during embryogenesis, elimination of self-reactive lymphocytes, also death of various cells after they finish their purpose. Example, neutrophils and lymphocytes after inflammation. The pathological causes include DNA damage, accumulation of misfolded proteins. This is also known as ER stress, and viral infected tissue. This morphology is very different from what is happening in necrosis. So here, normally you see a cell, it's a normal cell. In necrosis, there is swelling and formation of blebs. Here in apoptosis, there is cell shrinkage, and also there is formation of blebs. These blebs are formed to form apoptotic bodies later on. Which can be removed by macrophages, and there is no associated inflammation. There are two pathways which apoptosis takes place. That is the intrinsic pathway, also known as the mitochondrial pathway, and the extrinsic pathway, which is also known as the death receptor pathway. Ultimately, both of them lead to activation of caspases, which lead to activation of endonucleases and cytoskeletal and proteases, which causes cytoskeletal breakdown, formation of apoptotic bodies, which are phagocytosed by macrophages. Let's start with the intrinsic pathway. This is regulated by a family of proteins called as the BCL2 family. It contains three groups. The first one is the pro-apoptotic, also known as the BCL2 family effectors, essentially because they're causing apoptosis. The examples are BACs and BAC. Second group is the anti-apoptotic, also known as the BCL2 family regulators. Examples are BCL2, BCL-XL. And the third one is the sensors, BCL2 family sensors. They sense cell injury, and activate the effectors. So they sense growth factor withdrawal, they sense the DNA damage, either by radiation, toxins, or free radicals, and they can sense ER stress, that is increased protein misfolding. The examples of these sensors are BAD, BID, BIM, Puma, Noxa. What actually happens is the regulators, the BCL2 family regulators, which are present here between the inner and outer mitochondrial membrane, they maintain the membrane integrity. Of the mitochondria. What happens is when there is the cell injury, these BCL2 family sensors, these are also known as BH3 only proteins. They antagonize this regulators and activate the effectors, causing cytochrome and other pro-apoptotic proteins to leak out, especially the cytochrome C to leak out. This further activates the caspases. What further activates the caspases? These caspases are again of two types: initiator caspases and executioner caspases. The initiator caspases in the mitochondrial pathway are caspase 9. Okay, next, let's see the death receptor pathway. This is either be due to FAS and FAS ligand and FAS receptor. I'm sorry for the spelling mistakes. Receptor interaction, or TNF ligand with TNF receptor, which further causes activation of few adaptor proteins attached to the receptor, again causing activation of initiator caspases. But in the extrinsic pathway, the initiator caspases are different. These initiator caspases are they're different from the intrinsic pathway. These are caspase 8 and 10, both of which activate executioner caspases. From here, the pathway is common. The executioner caspases are caspase 3-6, which activate endonuclease and proteases. The endonuclease causes ordered condensation and fragmentation, nuclear fragmentation, and there is breakdown of cytoskeleton with formation of apoptotic bodies. These apoptotic bodies have some ligands for phagocytic receptors. These ligands are these are phosphatidylserine ligands, which can be recognized by a phagocyte and can be removed, essentially causing no inflammation. That's it, guys. We have covered all the important points of apoptosis. To end with, we have a MCQ. Do post your answers in the comments. Thank you and see you in another episode.